Okay, so our next talk is actually, it's live, but it's from somebody on Zoom. So I'm gonna present him. Dr. Alfredo Hidalgo Miranda. He's the Director of Research at the National Institute for Genomic Medicine in Mexico City. And he works primarily on researching genomic alterations in cancer tumors. However, as the Director of Research, he has actively participated in the creation and coordination of the work group tackling the COVID-19 pandemic at the National Institute of Genomic Medicine. And he has published more than 100 research articles and has been cited over 10,000 times in international literature. So give us just a few minutes until we set up the talk. Thank you.
Bueno, al parecer yo no los puedo escuchar a ustedes, pero ustedes sí a mí. Entonces, si gustan, podemos iniciar la presentación. Thank you very much for the invitation. I just wanted to check if you were able to listen to me. I don't think that's the case, but anyway, I will begin with the presentation. Thank you very much to the organizers for inviting me to this very interesting meeting. And today, I will be discussing with you the topic regarding how the National Institute of Genomic Medicine became prepared for the pandemic and how we faced it in the light that we are a National Institute of Health and our main objective is actually genomic medicine. So it was something somehow natural that we were part of this response that our federal government set up in order to face this pandemic. So basically, I just wanted to give you a little preview of what we are, a little bit of context. The National Institute of Genomic Medicine is part of the Secretary of Health. There are 13 different Mexican National Institutes of Health. Every one of them is devoted to a particular area of medical knowledge. And there are two of us which are not really, we don't have the fortune to deal with patients. We mostly are focused on research. One is the National Institute of Public Health, and the other institution, which is basically focused on research, is the National Institute of Genomic Medicine. The INMEGEN was created in 2004 as the 11th National Institute of Health in Mexico, and our objective is to apply genomic analysis tools in order to understand the genetic basis of common diseases. Of course, this taking into consideration the particular genetic background of the Mexican population. As you know, Mexicans, we are a result of different degrees of admixture between European and mainly Amerindian populations. So we really have to take this into account when we deal with aspects related to the development of molecular markers for disease. So one of our main objectives is this area of population genomics, which we have to take into account when we want to deal with genomic medicine in an admixed population. So we were working in different research areas, basically cancer, population genomics, diabetes, when the pandemic came out. So in March in 2020, thanks to an agreement that we developed with the government of Mexico City, we received a couple of grants which allowed us to establish and implement a SARS-CoV-2 diagnostic lab in what was our nucleic acid sequencing unit. We had to reconvert our sequencing unit to a lab that was able to carry out high throughput diagnosis of SARS-CoV-2 infection. This took place in March, and it took around 3 to 4 weeks to set up the lab. Fortunately, since we are an institution devoted to mainly molecular biology, we had all the things that we needed in order to perform the diagnosis of SARS-CoV-2. So we just put everything together in a single lab and began working with RT-PCR diagnosis in this lab. Of course, we had to deal with different things. For example, just to be able to hire more persons for this lab, but fortunately we were able to do that. And since March 2020, in coordination with Mexico City's government and the health systems of Mexico City, the lab has processed more than 500,000 RT-PCR tests, mainly from patients in Mexico City. So the institute became very soon the major testing facility in Mexico City. And we are proud that we can say that we helped our city's government in order to make a better decisions regarding how the pandemic was going, the infection rate, the positivity rate, and also later on, as we will see in the presentation, we also had a very important role in following how the virus was evolving thanks to our sequencing project. So as part of this effort, we have developed and validated the protocol for the detection of SARS-CoV-2, both using the normal nasopharyngeal tests, and also we developed a test that has been working, been used. We use saliva as an option for the detection of the virus. And this was a very useful situation because sometimes we also faced problems with the logistics. Some of the plastics that you need for the reactions and the reagents were a little bit short on Mexico. So having another option for testing the saliva worked very good. And it was a very interesting option that we have been using for a while, and it works very good. This was a necessity because at some point of the pandemic, we were not able to get the swabs. So it was something that came out of necessity, but it worked out really well, fortunately. And of course, we wouldn't be able to do this without the technological infrastructure that we already had in the lab and the institution. For this effort, automation was something fundamental. So we were able to make very good use of our systems automation units in order to prepare a large number of reactions for the PCR diagnosis. And later on, these also were used for the preparation of libraries for sequencing. Of course, this wouldn't have been possible without all the support from our staff. And for this particular response, we had to hire more people. At the peak of the pandemic, we were processing more than 400 samples, 4,000 samples a day. So we had three different shifts in the morning, in the evening, and even in the night. And in that way, we were able to process this amount of samples in a single day. Fortunately, right now, the numbers are going down. And now we are processing about 400 to 200 samples every day. But of course, we are able to ramp up these numbers if it's needed. Hopefully it won't be needed. But if it's needed, we will be able to ramp up again to those numbers if it's needed. No? And of course, since we have the access to the positive samples in our institution, they were being processed here for the detection. The next logical steps was to use these positive samples in order to set up a sequencing program, no, epidemiology program based on sequencing in order to analyze how the virus was evolving in Mexico City and how different variants of concern were rising in Mexico City and spreading in the population. So we began working with sequencing. We contributed our first SARS-CoV-2 whole genome sequence to GSAID on August 26, 2020. We initially used long-read sequencing following the Arctic network protocols and bioinformatic pipelines using Oxford Nanopore technology. This was a very useful technology because we were able to run small batches of samples since we were not prepared yet for a high volume sequencing. Beginning with the Minion from Oxford Nanopore was a good choice. And in that way, we were able to ramp up the number of sequences that we were dealing with. Afterwards, when we finally got everything to sequence in a more massive way, we switched to the Lumina platform, and we were the first institution in Mexico to implement the COVID-Sec kit that was developed by Lumina. This kit was initially set up for diagnosis, but since it's based on whole genome sequencing of the virus, then what we did was to prepare the libraries with a COVID-Sec kit on samples that we knew were positive with a certain CT threshold. And those are the ones that we are using for sequencing. All the samples that we are generating right now are being sequenced on an exec 550. And actually, this is an old slide, but we've been working with the NextSec 2000 for almost three months. So right now we are working with the NextSec 2000, and we are sequencing a higher number of samples with this setup. Since August 2020 to November 18, 2021, we have sequenced and deposited to GSAID. Actually, this is up to now. In GSAID, we have deposited more than 18,000 complete genomes in GSAID. And this represents around 32% of the total of samples that have been sequenced in Mexico. Most of them has been sequenced in Lumina, as I just said. But now we are also we also acquired recently the Gridiron from Oxford Nanopore, so we will be running also the Oxford Nanopore technology for sequencing if we don't achieve the number of samples that are needed to fill up a run from the Lumina sequencers. See, the samples that we have been sequencing have been collected mainly from Mexico City and the Mexico City metropolitan area, but we also have samples from other states of the country where sequencing information is limited. On average, we were sequencing 558 samples per week with the Lumina platform, and we can increase this number to more than 700 samples per week. Again, right now, since we are facing a shortage of positive samples, fortunately, these numbers are going down. And that's why we decided to get the Gridiron, because with the Gridiron, you can run smaller batches of samples, and it won't be necessary to wait for to get this number of samples for filling up the Lumina sequencer. So in that way, it won't be necessary to hold on the samples until we get a full run, and this will not impact our times on how long it will take to upload the samples to GSAID, which is another very important parameter if you really want to make use of this information for public health information and decision making. And this is the case now, for example, in Mexico, according to COVID-CG, Mexico is between the 25 countries with higher than seven cases sequenced per thousand of cases. And the median time of sequence data deposition in Mexico is 28 days. So this is not bad, but if we take the numbers from the INMEGEN, we were able to upload the sequencing information even in 18 days. From sample collection to data deposition in GSAID, this is a very good number compared with other countries, actually, and it's almost 10 days faster than the average in Mexico. This is, we think, a very important point, because that's exactly the focus of genetics surveillance epidemiology program, no? To really be able to generate sequencing data almost in real time, so that we really know which are the variants that are currently present in a population like Mexico City in a certain period of time. And also to be able to detect the presence of new variants or variant of concerns when they arrive to the city, and be able to do something with this information regarding public health policies. This is what we've been doing. This is an old graph, but as you can see, we were able to monitor how the variants of the virus became present throughout time. A particular variant of interest was this one in green, which was actually this in blue was the variant B11519. This was a very interesting variant that we first detected in Mexico City, and it became the dominant variant in Mexico City during February, March, and April. Actually, we described in a paper how this variant came out as the dominant, and how it was impacting the clinical manifestations of COVID. But then soon afterwards, when Delta came, Delta wiped out everything else and became the most relevant variant in Mexico City at that time. Now this has changed, and now we have Omicron, which is the variant circulating in Mexico City, as it is in all parts of the world. No, So we have published different types of papers describing the work that we've been doing here. Some of them was based on the saliva testing. Other was the description of the first case of Omicron that was detected in Mexico. It was sequenced here in Mexico City at the INMEGEN, and we described how it changed the panorama, the view of the presence of the variants in Mexico City. So together with this effort, we have been doing some research regarding a better understanding on how these variants are being spread in a population that is so big like Mexico City. This is just some parts of the paper where we describe this variant, the 519, and we did the different types of analysis in order to try to understand where it came from, what type of mutation it had, and what was the impact of this new potential variant in a clinical setting. Fortunately, it was not really that bad, and it was also that something that we learned very soon is that whenever you are beginning to understand a certain variant, the next is already there, and it was swapped out by Omicron. And we had to change everything to better understand what was the Omicron doing in Mexico City when it took over this variant that was very prevalent in Mexico City. Regarding the saliva work, this was something that where we did a paired analysis in more than 2,000 samples in order to test how it was comparing the saliva sample against the gold standard. So it was a very large cohort, and in this paper we described that it was a very useful tool in settings where either staff or resources were a little bit limited, and it worked very good. And we also did some pooling strategies that were also part of an effort in order to diminish the cost of testing when we were dealing with a very high number of samples per day. Who also worked on different developments like endpoint PCR, RT-PCR-based conservation landscape for SARS-CoV-2 detection. This paper was recently published, and it was just another option that together with the traditional RT-PCR, it can be used for the detection of SARS-CoV-2. So the experience that we have gained so far is that the establishment and setup of the diagnostic laboratory in-house here at the institution was a key decision for facing the current pandemic. If we had not been able to set up this lab here, it would be it would have been very difficult to have enough number of samples for sequencing, and we would not be able to help the Mexico City authorities in order to face this pandemic. So setting up the lab was really a good decision. And apart from providing an accurate view of the development of the pandemic in Mexico City, the lab also allowed us to have direct access to positive samples for genomic-based surveillance, as I just mentioned. So one of the main bottlenecks that extends the period of time when you collect a sample and you deposit the data of the sequence in GSAID is the time it takes from the diagnostic lab to send the samples for sequencing. So basically, we eliminated this waiting time because we already have these positive samples here, and it was just a matter of selecting those based on the quality of the RNA and also, of course, on the CT of the samples. So this really had a big impact in diminishing the time from sample collection to data deposition in GSAID. So the inclusion and adoption of different sequence platform was also a fundamental factor for efficient sequencing and data generation. If we were only dependent on a single platform that would allow, that would have to wait for a certain 300 number of samples to be gathered before each run, this will also have an impact both in costs and in time for sequencing. So the use of the Oxford Nanopore technology that allowed us to run smaller batches of samples was also a very interesting tool for us at the beginning. And now it's becoming also very important right now when the number of positive samples is diminishing. So I think for the next months, hopefully if the numbers keep going down, as we all hope, we will be using more Oxford Nanopore sequencing for this effort. And the flexibility of the sequencing platform allowed us to rapidly incorporate the COVID-sec test in our production pipeline. So the idea is that we were able to go to establish a flexible pipeline, work pipeline, either for the automated generation of libraries for both Illumina or for the Oxford Nano. So this flexibility allowed us to move in both ways and get the best of these two worlds. Of course, whole genome and viral sequencing genomic surveillance require a steady and constant flow of positive samples to be cost and time efficient. I think this was demonstrated by our experience here. Setting up the lab here was a key point in order to be able to maintain real-time data generation by sequencing. If this continuous flow of positive sample exists, the next challenge becomes efficiency in library production, and then it's very important to have automation of these processes in order to really work with a high number of samples. And this automation becomes very important both in the diagnostic setting and in the sequencing setting. A lot of labs were struggling to go from hundreds of samples to thousands of samples in the diagnostic setting, just because they were working manually. And if you work manually, then you will have problems either with human error or with time. So automation becomes really indispensable if you really want to process a high number of samples. And of course, as I mentioned, time from sample acquisition to the position of sequencing into public databases is a measurement of the efficiency of the surveillance program. And it's the only way we can have real-time data on viral evolution. So it is very important to take this point into account if you really want to make good use of the sequencing data for public health decision making. No? And of course, we really have to pay attention to all the steps of the pipeline, including the logistics and the chain supply of reagents, plastics, personnel, and budget to keep the pipeline going. There were times in the pandemic when we really thought we had to close the diagnostic lab because we didn't have access to the plastics needed for the RT-PCR. Fortunately, we were in very close contact with the vendors, and this didn't happen. So we had a constant, we had the lab working constantly, basically 24/7 during the peaks of the pandemic, and we didn't really have to close it because of a lack of reagents. So, but it's something that we really have to take into account when we face this kind of situation like a worldwide pandemic. So that will be all that I would like to present to you now. I really hope that the experience that we had in the National Institute of Genomic Medicine is useful, and I feel really proud that we were able to set up this lab, and of course the sequencing pipeline that in my view particularly, I think it helped Mexico City authorities to better face up this pandemic. Thank you very much.
Thank you. I'm not sure if Dr. Alfredo can hear us. So.
Yes, I can. I have a question here at the chat. Do you want me to answer? Can you hear me? Can you? Well, I think I can answer the question that is here in the chat.
Alfredo, can you hear us? Hello?
Hello? Well, if you can hear me in the chat, then I think we had a question here. What are the prospects for supporting other labs, perhaps in more remote regions, to make use of small batch sequencing and further improve the national picture? I wonder whether Cabana's fabulous network of bioinformatic trainers might be able to help there. Actually, yes, we had some ideas, basically with the Oxford Nanopore sequencing technology. This was a very good examples in other countries it's a platform that really doesn't require a lot of initial investment. So it's a good way to start. And it's something that can be set up quite easily compared with a bigger sequencing like the Lumina platform. One of the thoughts that we had is that maybe these labs can run their samples and gather their FASTQ data and try to set up a centralized node for the bioinformatic pipeline. And that's where Cabanas can be very helpful in these settings, so that we can have a centralized bioinformatic pipeline setup where someone, anywhere where they are, they can upload their data and get information regarding the variants that are circulating at a particular time in a region.
Questions? No?
I cannot hear you, but if you write the questions in the chat, I can answer. I have another question. Have you explored at Inmagen other alternatives such as sewage monitoring? Yes, actually, we are just about to begin a project together with one of our collaborators. They are very interested in sewage and in order to see both the positivity of COVID in residual water, particularly not from a city, but from certain offices, governmental offices, where they want to know if we can monitor the presence of the virus and the quantity of the virus in a certain amount of water. So to have somehow a quantitative measurement of the amount of virus that you can detect, and in this way try to predict if there is a new the presence of the virus, and we will have another infection coming now. So we are still working in this. We are setting up a protocol based on droplet RT-PCR. So I think this will be something that we will have results very soon, and it will be very helpful in order to monitor the presence of a new, how do you say, brotes, outbreaks present in a population in a certain region. The thing here is that we are not really quite sure, and that also might be a very good question for Cabanas. The bioinformatics needed in order to detect potential variants in this type of samples. I think in our experience, it will be something that it's like a mega pool of samples, and we will find only those variants that are the most common. It will become a problem in the depth of sequencing and the bioinformatics tools that we will be needing to detect, to really have a good idea of if we can detect variants in these types of samples.
Okay. Yeah, does anybody have another question or? Ian, please. I'll write it down as well.
I apologize for persisting with the point, but there seems to be a huge amount of sampling that's taking place for 2 or 3 decisions that need to be made. And I wonder whether sampling sewage is going to help you make decisions. And to me, that's the challenge. What's the best methodology for tracking this pandemic and different pandemics? And what's the cost effectiveness of the methodologies put in place?
I think that's an excellent question. I don't think, at least for the follow-up from now on, I think sewage and residual water monitoring might be an option. I don't think it will be the best one due to technical challenges. And I'm not quite sure the experience, the international experience shows that it is possible, but I'm not quite sure if this method will be enough to predict another outbreak. So I think monitoring open population will be still needed, not in the numbers that we saw during the full-fledged of the pandemic, but I think it will have to be kept at a certain point in order to be able to really monitor what's going on. The other thing is that in sewage, at least from what I remember, I read, sometimes you might be able to find some variants that are not really common in clinical samples. So that's another issue. We don't really know if this is related with the bioinformatics, with the types of samples, of it's something that it's really happening. So I think a combination of continued testing on particular populations, for example, in offices or in governmental offices or in hospitals, together with this sewage approaches might be useful, but I'm not really quite sure what will be the best answer to your question. Of course, keeping the diagnostic labs running and the sequencing lab running, it takes a lot of money. So it also makes sense to see when we will be able to how many samples we will have to sequence in order to answer the questions that we're doing. I think it's something that we are facing worldwide, and the information that we'll be gathering will soon help us to resolve these questions. But I think it's not a matter that is settled right now.
I'm not an expert, but could you not end up measuring COVID that has a competitive advantage or a comparative advantage in sewage if you go down that route? Is that a possibility? So actually, the where you're sampling from could skew your results.
I think so. I think so. At least from what I remember, I read, sewage sampling might skew your results, and it might not necessarily predict an outbreak. So it's a matter of, I don't think there's enough data still, at least from what I know, and I'm not really an expert in sewage monitoring, but at least from what I remember, it's an option to see if the virus is still there, more or less to see how much of it is still circulating. But I'm not quite sure if this will be enough to predict an outbreak, a clinical outbreak, because sometimes due to the clinical development of the disease, you might be shedding viruses from the intestinal tract that will not necessarily become a respiratory disease in some patients. So this is one of the main issues that are skewing the results, and it will be a little bit difficult to transpolate what you find in the sewage to predict a clinical outbreak defined by respiratory disease.
Thank you so much, Alfredo, for joining us virtually. And we'll move to lunch. Please let's thank all the speakers in this session.